in the chat as we get started today. We love seeing who's here watching. Throughout the day, we will be taking live questions from the audience. We do this using the Q&A panel, accessible from the bottom of your Zoom taskbar next to the chat window. After each presentation, we will be reviewing questions and the moderator will ask the presenter. So once you open the Q&A panel, you can click to type your question, and we ask that you begin your question with the speaker's name so we know to whom it's directed, and be sure to hit send. And we will be checking the YouTube live stream for questions as well. If you are having any connectivity issues today, we have a couple of helpful solutions for you. The first is you can view the event live stream on our YouTube if you're having Zoom issues. So we will send a YouTube live stream link in the chat. You can also find the YouTube link on the event details login page where you found the link to this symposium from alleninstitute.org, or you can just go straight to our YouTube channel at youtube.com/alleninstitute. And we are live on YouTube now. You are also welcome to call in from your phone for better audio, so splitting the audio and video bandwidth from your current machine. You find that in the audio settings panel and click switch to phone audio and you will get the call-in information. Here is a brief overview of today's agenda. Like the last 2 days, we'll hear from some researchers at the Allen Institute and researchers at other institutions who have used resources from the Allen Institute. Today, we're featuring the Common Coordinate Framework and then the Microns Explorer. If you did miss the talks from Monday and yesterday, they are all posted on our YouTube channel. You can watch those later. And as a reminder, at the end of our program today, we'll have 2 tutorials on how to use the resources that are featured in the symposium portion and learn about what the Allen Brain Map can do for your research. Today's tutorials will be on the Microns Explorer and the Common Coordinate Framework, and they will run concurrently. The tutorials are in separate Zoom webinars, which you can find on that same login details page from alleninstitute.org. And both tutorials will be recorded, so if you pick one, you will be able to watch a recording of the other one at a later date. Well, we hope you can stay for the entire program today. If you do have to step away, we will be posting a recording of the symposium talks as well on our YouTube. And now, since we're right at the top of the hour, I am going to pass it over to one of our moderators, Chair of the Next Generation Leaders Council, Keith Hengen.
Megan, thank you very much for the introduction. And I just want to quickly say I've very much enjoyed the last 2 days. I think that today is going to be equally exciting, and I appreciate everybody's efforts in assembling this and putting this together and delivering such wonderful talks. And I want to just take a quick minute to discuss the Next Generation Leaders Council or program and what that has sort of meant for me and some of the other people on that council, and to encourage people who are eligible to apply for that. And so before I dive into that, I would like to introduce the incoming chair, one of them. So Tina Kim and Eric Ittrey are the rising chairs of the Next Gen Leaders Program. And so Eric is going to be the moderator of this symposium today. Eric, can you unmute and join me?
Here I am. My name's Eric Ittrey. I'm an assistant professor at Carnegie Mellon University. And yeah, I'm excited about today. And I've got to say that the NGL program has been really special. It's not just an award. The community and the opportunities that have been provided through it, and the opportunity to influence science a little bit and see how it works at a large scale has been pretty fantastic. I know in my experience.
Yeah, so just very quickly, so if you are a senior postdoc or you're just starting your own independent lab, you're eligible to apply to be a next generation leader. And there are a lot of sort of private foundation awards, and this should not be confused with that, as Eric brings up. So there's no sort of monetary incentive for this, but you actually play a guiding role on the scientific leadership of the Allen Institute. You kind of get to sit at effectively a kid's table. So there's a very active role. You head out to the Allen Institute twice a year, and you get to really embed yourself in the science and meet all the scientists and everybody who makes the Allen what the Allen is. And the last 3 years, I would say that being an NGL is the single greatest impact on my career of anything that I've done. I've grown grants out of the relationships I've built there, working on papers with people there, and I have a whole new community that I would not have had otherwise. And I struggle to imagine where my lab would be pointed today had I not been so engaged with the Allen Institute over the last few years. I mean, it's been truly an honor and an amazing experience, and so I cannot recommend it strongly enough to people. I don't know, Eric. Maybe you'll give like a slightly more moderated version of that, but it's been fantastic. And I think Tom would say the same thing, but I think I'm not sure he's here with us yet. But so Eric, with that, is there anything else you want to say about the NGL, or should we let you set up the day?
I think you did a pretty great job. Awesome. If anyone listening has any questions about the program, I'll put my email and everyone else's into the chat. Please feel free to reach out to us. As Keith said, it's been a pretty fantastic experience. I'm looking forward to sharing in this coming year.
Yeah, so I want to make one last point on this, and then I'll leave it alone. I think that the selection for the NGL, a lot of people reached out to us in this past year, sort of saying, does my stuff fit? Am I eligible for this? And I think that there's a real focus on diversity in this, and I mean diversity in terms of intellectual approach, the type of position people are in, the types of things they work on, because my instinct is that the folks at the Allen really want intelligent people who are looking through diverse lenses to be able to comment on the future of their work and the ongoing work. And so feel free to reach out to us if there is any question of your fit or potential eligibility, and we can sort of at least give you our perspective on that. Okay, Eric, over to you.
Thanks so much, and welcome back everyone to the last day of the Open for Neuroscience Symposium. Today we're featuring two projects. First is the CCF, or Common Coordinate Framework, of the mouse brain anatomy projects, and then the Microns Explorer that really focuses on electron microscopy data. But before we get started with that, I wanted to highlight some other opportunities at the Allen. If we could put the links up on the screen and share that slide. Excellent. So there's a summer workshop this summer. If you're interested in learning more about the Allen Brain Observatory and are a graduate student or postdoc, particularly with a systems or computational bent to your work, it's a pretty fantastic opportunity. This is something you have to apply for, and the deadline is March 25th. And so that'll be an exciting opportunity for some of the more junior scientists out there. The Institute has also just opened up applications for a new fellowship. Important here, it's a fellowship, not a postdoc, but it's at the intersection of data science and neuroscience. And this is the Shanahan Family Fellowships that are designed for people transitioning from that data science and math side into neuroscience. And I know my own work and work that we'll see today, that's ripe for opportunity. You can also apply to the next class of the Next Generation Leadership Council. Myself, Keith, and Tom are chairs, as well as Tina. We're the incoming, and Keith and Tom are the outgoing. And again, please reach out if you have any questions about that. And that's open to senior postdocs as well as early career faculty. And there are lots of other open events like the one that we're in right now. We hope that you join us for that. And as we begin this day, I'd like to take one more opportunity to thank the late Allen Institute founder, Paul G. Allen, without whom this institute and this event and much of the research you see would not be possible. And if you're interested in joining the team yourself, there are open positions available, ranging from research associates to software engineers and investigators and everyone in between. So with that, I'd like to get started with today's talks. First of which will be an overview of the mouse brain common coordinate framework. And later today, of course, the Microns Explorer. We're going to start each section with presentations by Allen Institute scientists who are using data that is also publicly available. And so being open is an important theme for the Allen, and certainly that's been highlighted in the past few days. Following that, we'll hear from researchers at other institutions who are exemplars in using that data and making some really important advancements. So the Common Coordinate Framework, or CCF, is an anatomical framework that underlies many of the Institute's resources, many that we've heard about already. To me, it's both very beautiful as well as impressive, and it serves as a foundation for so many tools. Our first speaker is Lydia Ng, an investigator in the Allen Institute for Brain Science. Lydia will also be presenting a tutorial on CCF at the end of today's talks. So please make note of that option for the tutorials at 11 o'clock Pacific time for that. So with that, take it away, Lydia.
Thank you, Eric, for that lovely introduction, and to all the organizers for inviting me to speak. So to start, good morning, afternoon, evening to everybody listening in. I'm Lydia Ng, investigator in Allen Institute, and I'll be talking about the Allen Common Coordinate Framework as a 3D reference atlas to enable data integration. Just getting used to the slide kicking. Okay, so why do we need a common coordinate framework? So in the first 10 years of the Allen Institute, we generated a lot of imaging data. Our first project, the Allen Brain Atlas, was gene expression data over the whole mouse brain for over the whole genome. That was 500,000 images. So we needed a reference atlas and eventually a coordinate system in order to integrate all that data. Moving on from that, we did the developing mouse brain atlas. That was more gene expression data, yet another 500 million images. And then to support that project, we developed the Allen Developing Mouse Reference Atlases. After that project came the Allen Mouse Connectivity Atlas. That brought new challenges and also new opportunity, yet another 500,000 images that we needed to integrate. So the Allen Common Coordinate Framework was born out of the connectivity data. We will describe that more in detail in just a moment. Moving on, our second decade at the Allen Institute, we are more looking at integration of single cell data. As a technologist, single cell data is code for anything goes. So as we build out this new Atlas framework, we needed to have something that could integrate a lot of different multimodality data arising from many different assays. Moving beyond what we do at the Allen Institute, we're also part of the Brain Initiative Self-Census Network, BICCN. Here it is more than 30, 40 different individual research groups, all looking at the cell types of the mouse, the human, and non-human primate. Our team plays an important role, the blue box in the middle, which is a coordinating center. And one of our charter is to integrate data both in the spatial and semantic framework. And the Allen Common Coordinate Framework plays a role in that spatial integration. When we start off with, this is a picture from the paper that came out last year, and it's a great summary of all the different parts of how the Allen Common Coordinate Framework, the CCF, was constructed. In summary, the CCF is, the basis of the CCF is this sort of novel, as Eric says, very sort of unique and beautiful thing that we made, in which it's a 3D average of over 1,675 mice. This is where these mice came from, the Connectivity Atlas project. I don't think we would have an opportunity if we didn't have such a rich data set such as that. It is at 10 micron voxel resolution in 3D, allowing you to see exquisite details of brain anatomy. After we constructed this brain, there was a 3-year effort by our anatomy team to annotate it. And to me, annotation is like adding existing knowledge from the neuroscience community into this framework. We ended up with 43 cortical areas, along with their layers, another 329 subcortical areas, and then also the ventricular structure and also the fiber tracts, which is important to understanding the brain. Everything we made has its open access and can be available through that link, atlas.brain-map.org. All right, here I was just showing that all the names of everybody involved, because it took a village and practically 4 years to complete what you see out in the web applications. All right, so let's go back in time. Allen Mouse Connectivity Atlas. Briefly, this is a data set of projection data. Here, if you open up the atlas, there's a picture and there's all these little dots. Every dot represents an injection into a particular mouse brain. That injection has a virus load, and when cells are infected, it will make that entire cell glow green. And then we take pictures of it, and that picture represents the projection from that area to the rest of the brain. What was very special about this project was the use of a serial 2-photon tomography data, serial 2-photon tomography system, in which the brain is imaged the brain is imaged while it's still intact, and is imaged just below the surface. So when we get our data on the other side, we have these data set that is already in a 3-dimensional framework, free from wrinkles and tears that you'll get, that you would ordinarily get if you had an experiment which required cutting out the section and then laying it on a slide. So we had this rich opportunity of having many thousands of these rich 3D data to build a new atlas from. This slide sort of shows how the average template was built, or some iteration of it. So starting with, as I said, we started with the Allen Reference Atlas, and we made a 3D framework out of that. Starting with that as a template, what we did was register every one of our 1,675 brains to that, and then in an affine manner. And then you get, and once you do that registration, you average it. And then this, while it's fuzzy, becomes the new template. You do that again until you reach some more convergence. What's also special that we do as we get a more clearer template is that we also want to make sure that it's also a shape average. So at each step, after we do a registration, we also average the deformation field to get to that space, average it, invert it, and then apply it to the brain, to the template as well. At the end of this iteration, you'll end up with a 3D volume that represents an unbiased intensity of that population, and also an unbiased shape. As you can see, as you go through the different iteration, more and more signature of the anatomy and features become sharper and sharper. in the box here, H here, you can see the barrels of the cortex. In the middle row, we did a sort of a maximum prediction intensity over 3 views of the data so that you can see a lot of the signatures, hallmark of somatosensory cortex. The cerebellum can be highly, parts of the cerebellum can be obviously can be seen. And in the last row, as you zoom in, you will get, you will see more and more detailed features. Here is a zoom in version of that. As I said, that in a single brain, when you look at single brain data, the barrels are there, but they're kind of fuzzy because we have so many specimens allowing us to take like different views of it. In the average, these things become very crisp and clear. Since we're talking about barrels, there's these barrelettes at the medulla of the brain. Again, in a single experiment, it's very hard to see where they are, but over the population, the signature pops out so that you can actually see the whole barrel fill in the back part of the medulla. Same again in the thalamus. Here in single brain, you can't really tell what's going on here, but in the average, the stripes that are associated with the barrel fill can be seen. This makes this average template in itself a great anatomical resource. And I personally probably spend too much time sort of looking at it and looking at what does those shapes mean, because they're highly unusual. But typically, if you go back and look at what's in the literature, you start to see how things that you see in schematics are actually being shown in the average template. Okay. Now that we've got this gorgeous 3D average template, came the hard work of annotating it. So for every structure, our anatomy team starts off doing research on that structure. What have people said about the structure in the literature? What does it look like in the current atlases we have today? And then they will use multimodality data, transgenic characterization, histochemistry data, ISH data, gene expression data, all to help confirm these structures in the brain. Annotating data in 3D is kind of time intensive and cumbersome. So there's yet a second workflow in which our senior neuroanatomists will go in and do primary delineation in a subset of the images. And then our research assistant team will come in and then help fill in that 3D shape by at every step trying to make sure that the shape stays a 3D integral shape. And then even then when you've done this for a nucleus, a set of nucleus, at the end of the day, everything needs to fit together. So there's a refinement step to make sure everything is adjacent to each other if they need to be in the final 3D product. All right, so this slide is just to show some of the exquisite details that our anatomist team went into annotation. Here is one figure from our paper in which the interpeduncular nucleus was highly sectioned into smaller sections that is new. And then also have this 3D integrity. There's a lot more other vignettes in the paper as well. And if you come to our tutorial, there's a couple more in which we haven't annotated them, but you can see it in the average template. Okay, but when it came to the cortex, and because this was not the first time we did a 3D framework, we wanted to do something special in the cortex because we knew that a lot of our cell types data will be in the cortex. So what we did was then we created a curve coordinates because we know that the cortex is a sheet, it's a curved sheet, and that there is a columnar architecture in this sheet, such that single cells are apical dendrites, and yet when cells communicate with each other in the cortex, you can see that they do this in this sort of columnar organization. So when we came to look at the cortex, we want to actually build that in as part of the CCF, and to make sure any data that we're looking at or any integration do obey some sort of columnar framework. So in order to make such a framework, first thing we did was to identify the isocortex, which was done by looking at the average template and also using a couple of Cree lines. Then we did some mathematical magic in which we solved the Laplacian to make these sort of equidistance potential surfaces between the two surfaces. That's what that rainbow represents. And then once we have that, for every point in the surface, we did sort of a steepest descent path from the pier to the white matter. And then all of these sort of lines that you're seeing in F shows that the set of streamlines we're able to get for that. So this set of streamline is then approximates our columns, and this is what we use to integrate data and also to draw isocortical areas. So once we have these streamlines, it is then possible to project data to the surface, making it easier for our anatomy team to see all the information in a single sort of 2D view. So the top row here, you see that we've projected all the, just the average intensity from the template. And you see that just on the template alone, you see all the hallmarks of the primary cortical areas. Here, the triangular shape of the visual cortex, the barrel field, and other domains of the somatosensory, and also and also highlighting the auditory cortex. In addition to the average template, we were able to project Cree line data also to the surface. And then, here shows us being able to integrate connectivity data. The colors, the different colors here are different visual injections into the primary visual cortex. They're all integrated and color-coded to help us look at sort of the way that the visual field is projected into secondary visual areas, and then with all of this information, then our anatomy team were able to then determine very precise boundaries in the cortex. The streamline also allows to do some computation about the cortex as well. In the bottom row, now instead of just looking at from the top, we had flattened out the cortex. We were able to show where, in this top one, which direction the streamline was facing. Is it facing front ways, anterior, or was facing posterior, lateral, or medial? We can measure the different thicknesses along the cortex, whether or not the streamline is facing up or facing down, and also projects these lines from a top surface view into a flattened view. Keep an eye on this purple dot here. Here we know is a position in the somatosensory. We know that it's facing forward and it's facing up, while in the green here, in the visual area, it's actually facing more laterally, and then it's even further upright than in that area. Because once we have these streamlines, we wanted it to confirm it with real data. So again, in our projection data sets, we had information about these thick-tufted layer 5 neurons from our SIM1 Cree KJ18 Cree lines. Here in this injection, you can see that while the injection was in this plane of coronal plane, you don't see the apical dendrite. It's tilted forward, so you have to go back anteriorly to actually see the apical dendrite, and then you have to go posteriorly to see its connection to a pier surface. So in a sagittal view of this data, you should see this lean, which we see here. So in this next figure, what we did then, what would it look like if we flatten the whole thing out, and what does the data look like? If all goes well, everything will be pointing upright. So in our big purple spot here with the somatosensory, this is now our new flattened coronal view, and this is now our new flattened sagittal view. So this these three different sort of injection sites or affected cells, we can see that now indeed we have straightened everything, and we see the all the apical facing up, and we see all the projections down to the pier, the white matter cells facing down. And then in this view, these sort of three different blobs have become very clear as well, because they are now they now have been put unbent. In the visual cortex, you would see that it's already very upright, because that part of the brain is it was very lateral, so it was already facing in a sort of straightened direction, and that and this also confirms that we have our streamline is following the curvature of the cortex. And then the front of the brain, this is where when you look at the data, you don't see the tufts at all, because the cells are practically sitting lying down and going front to back. So that while it's a little bit more fuzzy, after you straighten everything, you do see the apical dendrite facing up in both views. So we were happy with that. And so and then using that framework, so now we have our 3D template. We have now annotated all the data. After 3-year process, we start integrating this into our product. First thing, you can see everything. You can see the CCF. We use the CCF by going to atlasbraindash.org. You can also view the data in 3D in our 3D viewer, and also look at it in the 2D interactive atlas. All right. Here, as well as integration of this product, we are also using it within the BICCN framework. Here is just a sort of a teaser of some of the work we're doing. Here on the right here, these sort of segments here you see in this, that's mapped to the CCF, shows all the brain areas that are being dissected by the ECHA team. They have a U19 in the BICCN, and from those dissection, they generate lots of transcriptomic and epigenomic data. And they like to do this through like a big dissection. At the Allen Institute, we do sort of a laser micro dissection in which we generate much smaller sections, and then we do single cell transcriptomic for those. Even though that these two methods are very different, by integrating everything in the CCF, we're able to compare and contrast the location of the single cell, and then also add to its interpretation if we see gradients and special signatures in the transcriptomic data itself. All right, we have done this with our multimodality data, in which our data, our ePhys, morphology, and transcriptomic data, all in a single cell, are pinned to the CCF, so that can be integrated with other data. And then because the CCF is available for open access, it has been used in the community already. We have seen it being used to sort of interpret and compare wide field data. And so that top view, which we knew was important, has come into play. It has been used to compare against functional data, if fMRI. So then the 3D volume was helpful to then connect to other 3D volumetric data. There's this lovely tool called Brain Renderer that allows people to make 3D visualizations of different anatomical areas that has been used in many different people's papers. People have built registration and segmentation tools, and also using registered data to see where there are connectivity, big connectivity patterns that the brain shows. All right. So with that, I'm going to finish up. And first of all, I would like to thank our founder for his vision and encouragement and support, without whom none of this work would have come to fruition.
Excellent. Thank you so much, Lydia. I wanted to start off a round of questions, and if you do have questions, please put them in the question and answer, and we'll get to them. Our first question comes from Heidi Clevin, and the question is the Allen Institute Brain CCF version 3 the final Atlas version, or will there be new iterations in the near future?
There will always be a new iteration of anatomical atlases. But I think one of the things that we want to do in the future to make sure that we sort of everything is integrated when you do add a new boundary, saying where it's from and what do we know about it. And it's probably atlases of the future would probably be many different layers and maybe different interpretations. So yes, another iteration, and looking forward to be sort of the type of resource that we need to integrate data.
Excellent. And along those lines, I actually was wondering, atlases have existed for decades. In developing the CCF, were there any noticeable differences you found from this to prior similar tools? Or more importantly, if you wanted to highlight maybe the greatest advantage over this versus the more standard atlas?
I am going to highlight its greatest advantage, and also its greatest controversy was the isocortex, because we wanted to make sure everything was drawn on a curve and everything was perpendicular at every angle. That meant that when you look at the coronal section, things looked funny. And tradition, we have a lot of controversy. Why does it look funny? Is it real? And because when you're doing a traditional atlas, it's hard for you to think about a perpendicular in a 3D dimension. You will tend to go in this coronal section, perpendicular is here, and I'll draw my boundary here. So that was a new highlight and a new controversy. But I think something like a true 3D sort of perpendicular is needed. So this is ways in which we all learn from what we know, and we can sort of then integrate what we know from the past into the future.
Excellent. And would there ever be an option to go between the more traditional version and the flat map curve projection?
We have, because we base it on coordinates, we can always map any atlas that has been registered back in the framework. So if you look at our downloads, you can also actually get the 3D delineation from the previous atlas that can overlay on the CCF as well. So it is one of the layers that we make available.
Excellent. Thanks so much. And I guess my last question would be, I mean, this is incredible power. I'm looking forward to seeing the workshop. Do you happen to have a dream experiment that you'd like to see this tool used for? If you could see it used any way, what might you want to see happen with it?
I think my dream experiment is because there's a lot of, okay, so if you come to a workshop, we see a lot of lines. We see a lot of lines in the CCF that we don't see, like if you look at traditional histology, which is cell-based. So my dream is to interpret those lines and how do those lines, they have to do with development. And then how do we then interpret those lines in terms of development such that we can link it to other species? That's the dream.
Excellent. Well, thank you so much for an exciting talk. And next, we'll hear about how the CCF can be used in research, sort of a good leeway. In this case, with 2-photon imaging data, much like that in the Allen Brain Observatory. Bingwen Brunton is an associate professor of biology and the H. Stewart Packer Faculty Fellow at the University of Washington. And she will be presenting Go with the Flow, visualizing spatial temporal dynamics in optical wide field calcium imaging. Thank you, Bing.
Thanks, Eric, for the introduction, and thank you for the organizers for inviting me here and for the opportunity to share some of our research in the symposium. I think what I'm talking about is but a very small example of the beautiful work that Lydia just introduced with the Common Coordinate Framework. And it's something that is it's a new direction for my lab, so I was pretty excited to have the opportunity to share it with you all. So before I jump into this particular project, I wanted to, I'm waiting to see if the slide advances. There it goes. Thank you. So I wanted to just say one word about what my lab is focused on. So things that we're really excited about are data-intensive methods to understand neurocomputations, especially understand underlying naturalistic behavior. So a lot of students and postdocs in my lab are focused on different model organisms and their natural behavior. And we have projects ranging from insects to rodents, non-human primates to humans. So on the right-hand side, you see a picture of a human. She happens to be my daughter in her natural habitat doing the behavior that she loves to do best, which is eating spaghetti carbonara. I don't actually study her, but I needed a picture of a human, so I had to put her up there. But that's not the type of data we're talking about today. We're talking about a particular type of data that I think many people here are familiar with and that Lydia has already mentioned in the previous talk. And it is wide field optical imaging data from GCamp in mice. So in this particular data set, I'll just tell you the story of this collaboration. And I think it's been, like I said, a new direction for my lab. And I was really excited when I ran into my downstairs neighbor in University of Washington, Bill Moody, and his graduate student, Dennis Tabuena, who has been taking these wide field calcium imaging data sets of mice during development. So these are pups from 1 day to, I think they did up to 10 or 11 days after they had been born. And when we looked at this data set, one of the things that was really jumping out to us, and I think jumps out to many people who look at these types of data sets, is that there is, for lack of a better word, flow of calcium activity in the cortex. So here we're looking at is a particular pup. I think this is a P7 pup. The dashed line on the top is the midline, so kind of looking at the left half of the developing cortex. And what you see, first of all, is that, well, there's a lot of activity, but most particularly, if you watch for a little while, you'll see that there are bouts of these waves that tend to originate from a couple of different locations, and then they grow and propagate over large swath of cortex, sometimes taking over the entire cortex. And so what we wanted to do was come up with a way of visualizing and quantifying this type of movie information in the way that captures the flow of calcium activity in the mouse brain, because it's really hard to capture it without watching these videos. And as much as I love watching videos, you can't so far put a video in a PDF of a paper you're writing. And so we are still dependent on ways of summarizing spatial temporal information into flat projections so that we can actually put it in a paper and also summarize it across many, many examples. So the features of this type of data that we wanted to capture, again, are spatial temporal flow. So not just static spots of brain where there's activity and there's not, but rather that the activity seems to propagating these waves, right? And so these translating waves is a particularly challenging problem. And this is where we were inspired by taking methods from a very different field where all they study is flow of fluids. The project turns out to have been led by a very talented undergraduate student in my lab at the time, Nate Linden. Nate has since moved on to a PhD program at UCSD, so we're very, very proud of him for this progression. And so what Nate did was, so here's a video that Nate took while he was out hiking in the Sierras. And this is something that we see in nature all the time. So for example, I saw it this morning when I poured a little bit of half and half in my coffee, because you'll see the swirls as you mix two types of liquids together, right? So it's very intuitive for humans to think about mixing of flows. What you see here in this video that Nate took of this little brook is that if I move my cursor, can people see it?
Yes.
Thank you. So what you'll see here is that there's a line here where on the in the middle of the stream, the water is going really quickly, and closer to us, because there's this giant boulder here, the water is kind of stagnant. It's not moving as fast, right? So that's the intuition that we're after here. So how can we use methods that are typically used to characterize fluid flow, like the kind that you might see going hiking, and looking at rivers, and when you are mixing your coffee together in the morning, how we use those methods to capture the flow of calcium activity in mouse cortex. And it turns out that these methods do exist, and they've existed for a long time. And so I'm going to introduce the set of methods here. And it's a little hard to gain intuition. So a lot of what I'll do in this brief presentation is trying to gain a little more intuition about what these analytic methods actually tell you. And one way of thinking about what they tell you is they tell you about material accumulation lines. These material accumulation lines are really, really common, and you can visualize them even from space. So here's a satellite imagery of clouds moving past a particular island. And so here the wind is going from the top left towards the bottom right. And you'll see the very canonical, beautiful von Karman vortices that shed off of this mountain in the island. And so these swirls here, these lines where the cloud particles tend to accumulate, those are the lines that we're after. These are material accumulation lines. So if you didn't happen to have a bunch of clouds and some wind to visualize this, because we don't have that in the mouse cortex, the way that we do this is going to be a method that's a bit of a mouthful. It's called finite time Lyapunov exponents, FTLEs. And I'll just contrast that before I start telling you what it is with more traditional modal decomposition ways of analyzing this type of data, the kind of thing that is very much my first impulse when we started analyzing these types of data, and very commonly used to analyze wide field calcium imaging data. And so the methods, and of course there's many more versions of there's more bespoke versions of each of these methods, but we're looking at modal decomposition by something like PCA or SVD, or something like non-negative matrix factorization. And so both of those methods and derivatives thereof are really excellent for reconstructing the data in terms of static modes. And they work really well in that context. But what they particularly fail to give us is the intuition of, like we looked at the movie earlier, the translating wavefronts and how the calcium activity flows through cortex. So that's what we're captured here with what we are calling the flow portrait method. Okay. And I know these are just lines with colors and they don't make any sense yet, but again, I'm going to try to build up a little more intuition. So the method fundamentally is a spatial temporal and analytic method. And so unlike PCA and non-negative matrix factorization, the first step is extracting a vector field from the data set. So here we are using a tweaky version of an optical method. So turning a movie of wide field calcium imaging data into a vector field. And this is time varying vector field, as you will see. The next thing that we do is we computationally plop down a equally distant spaced grid of particles, of virtual particles. So this is the analogy of the cloud particles flowing past Guadalupe Island that we saw earlier in the fluid visualization. And we let those virtual particles flow according to the vector field that had been extracted in the previous slide. So this is going to play on a little loop here, just so you can kind of see it, a tiny snippet of it several different times. And what you'll see is that even though the particles started out in an equal distant grid, once you let them flow according to the vector field, some of them will get bunched up together and some of them will be repelled from each other. And that's what we're after here. We're after the areas of maximum repelling, so those ridges where material is maximally repelled according to the vector field. And if you do that in backwards time, you'll also get areas of accumulation. Formally speaking, the finite time Lyapunov exponent is the largest eigenvalue of the Cauchy-Green deformation tensor. The details don't matter for this particular talk, but I wanted to define it to connect to that very extensive literature in the analysis of fluids. And so if anyone has any experience with that, this is almost exactly the same thing. We've had to make a couple of different tweaks to the algorithm for the fact that calcium flow in cortex is not incompressible. So there are some assumptions we had to relax, but it's essentially the same intuition there as well. And what you end up with is two maps. There is the forward FTLE map and the reverse FTLE map. The forward FTLE map is if you do the method that I just suggested by flowing the particles through this little vector field, and you do that in forwards time. And if you do the same thing, but playing the movie in backwards time, you'll get the reverse FTLE as well. And here's what those ridge lines would look like if you had done that. So that gives you what we call the flow portrait. And the flow portrait has forwards time FTLE, which are, you can think of it kind of as the sources of activity, where the activity is coming from. And there's also the backwards FTLE, which you can think of as sinks, where the activity goes to die. It's slightly more complicated than that because you also get wave fronts, which are not well summarized by describing them as either sources or sinks. But I think this is usually a pretty good start in terms of intuition. So we have sources and we have sinks. You might be tempted, if you remember a bit from your vector calculus, to ask how this connects with just computing the divergence of the vector field. It's similar to that, but it's also a bit different. And that's something that I'm not going to show you right now, but it's something that we also investigated exactly how this is different for if you had done that analytic approach. So before I show you more results from actual data, we are going to just look at a really simple synthetic example of a Gaussian that pops up, translates, and then that activity goes away. I'm going to play this movie here. So it's going to loop a couple of different times. And if you look at the movie on the left-hand side, you'll see the synthetic data set that Nate made up. So you'll see that we knew the right answer for this one. It's just a Gaussian. It's a Gaussian that pops up, translates, and moves over. And what you'll see is that you have the forwards and the backwards FTLE lines in purple and in orange. And what I'm showing you is what those lines look like depending on different integration lengths. So what is the window of vector fields that we extract? Okay. And I'm showing you here that all of these different integration lengths partially as a way of illustrating that is another, I'm just being honest about hyperparameters, right? That is a hyperparameter in this algorithm. And if you choose the correct integration length, things look sensible because this is a very symmetric movie, right? This movie is exactly the same if you played it backwards and forwards. And if you don't choose the correct integration length, if you don't choose a nice integration length, then the lines don't make as much sense. And so that's a little finicky, right? So user beware, if this is something that you may be interested in doing for your data set, we do give some guidance in the preprint about how to pick these integration lengths, but it is rather important to pick one that is correct. This is not uncommon for analytic methods of complex spatial temporal data. So for in full disclosure, that is a parameter in our algorithm. So we applied this to the motivating data set from Bill Moody's lab. These are developing pups. So here is one movie again from the mouse pup. This is a P7 mouse. And you'll see in the case of this particular movie, there is a spot of activity that originates around here where my cursor is, grows and gets larger and translates a little bit to the upper right. If we apply the flow portrait method to a bunch of different movies, here we're showing you each row is a mouse going from P1 to P8. And then each column is just a separate sleep bout. So these kinds of waves in developing mice tend to happen a lot during sleep. So we have flow portraits of yeah. So this is just this is a one image summary of a tremendous amount of video data. Right. So each of these little visualizations is actually a small movie with rotating, translating activity that we've then compressed into a small picture. So the science of exactly what this means is something that we've been discussing with Bill and with Dennis, but I'll just say that kind of in at least in colloquial terms, one thing that we've noticed is that the coherence of these FTLE lines differ throughout development. In the beginning, they're a little bit speckly. I know that's not a scientific word, but in the very young pups, they're a bit speckly and they only become a bit more coherent. The lines are more connected as the pups get a little bit older. And we also applied this to an adult mouse data set. This data set was collected by Nick Steinmetz, who is also a next generation leader, or at least was the past one. I lost track of exactly where he is on the council. So this is an adult mouse data set where it's almost exactly the same type of data, but we're looking at a top-down view. Interior is up, midline is in the middle, and you'll see that this is actually spontaneous activity in the adult mouse. The mouse is just hanging out in the dark, running on the ball occasionally, and that's all they're doing right now. And what you'll see is that these flow portraits meant very little until we were able to overlay them on the Allen Common Coordinate Framework, which case we can then actually correlate the sources and the sinks of activity that we were seeing over many examples and look at where they are relative to known anatomical landmarks on the adult mouse. So I say this as just one small example of an application of the Allen Common Coordinate Framework that Lydia introduced in the last talk. Rather than a complete story, this is very much just the beginning of something. I think this visualization technique captures some features of this type of data that is becoming more popular. I feel like lots of people are starting to collect this type of data set. And I hoped that this will be another way of capturing the spatial temporal dynamics in this type of data set. And the ability to overlay it on top of the Allen Coordinate Framework has been really useful for us in terms of connecting what the lines mean relative to the underlying neurobiology. And so that's been really powerful in our work. So I'll end by saying that I told you what I think I do, which is data-driven dynamic models of brain and behavior. What my kids think I do is, this is a little book that came home, my son, when he was in kindergarten. My mom and I like to do math, and my mom really likes brains. So my kids know me pretty well. This pretty much sums it up. What I really do is I have the opportunity to work with a really wonderful group of students and postdocs. And I only was able to show you the work of one of them today with you today. And that's a shame. And this is a picture from 2019, which is basically the last time we were able to gather in person as a group. And so I miss them, and thank you for your attention.
Thank you so much, Bing. That was really, really exciting. We had a question from Keith, one of the panelists. Keith, do you want to go ahead and ask?
Sure. Bing, first up, I love that closing slide, the drawing from your kids. It's very, very touching.
Thanks.
So this is maybe more of a take this as an open-ended question. It's a little bit rambling. So I typed this out as you were presenting this. I said, the fundamental characteristics of fluid dynamics. Oh, my question just disappeared. Okay, hold on, let me find it again. Okay, the fundamental characteristics of fluid dynamics seem to be highly dependent on the fact that particles only interact with their immediate neighbors, i.e. something like a nearest neighbors topology. So how do different topologies that include more long-range connections fit with these flow-based methods? Does this introduce discontinuities in the flow portrait? And does the fact that you do get such a nice flow portrait impose constraints on our understanding of ground truth topology in the brain?
Excellent question, Keith. Very insightful, as I expected from you.
Thank you.
So absolutely, this is the underlying assumption that we made in this iteration of the flow portrait. And it's something that we've been thinking about relaxing in certain ways, because in fact, this neighboring topology, the fact that particles only interact with their very near neighbors, is essentially a graph on the cortex, right? And so, as we know, cortical areas do connect to other cortical areas that are long and long-ranging through other pipes that are not in the neighbors. And so that's it's a different graph. And then lots of other people, including a couple of folks in my lab, have tried learning those graphs, like the connectivity, not only of cortical areas to their immediate neighbors, but to far away other cortical areas that are also observable with calcium imaging. And so how you would adapt this onto a flow portrait would be that, I think it would basically tweak the way that you would infer those little arrows, right? You would basically infer a slightly different vector field to admit the fact that activity might teleport from one spot to another. We haven't done that yet, and I think it won't be easy, but I don't think it's categorically impossible. But yes, you're correct. This is an assumption you would make.
I think that's awesome. I hope you guys are able to solve that problem, because it's
Me too.
cool.
But yeah, you're right. Like it does approximately work, right? At least in some of it. I think it worked a little. I think this assumption may be slightly more accurate in some of the developing pup work that we've done rather than the adult mouse. I don't really know what's going to happen when they're doing a task, right? So all of the activity we've studied so far has been spontaneous.
It certainly suggests that whatever calcium is indicating, right? And we got into a discussion about this yesterday, that calcium is more than just spiking. So whatever these kind of large scale calcium events are, really do tend to move this way. And that they do have a very kind of fluid dynamic local propagation basis. But I think it would be awesome if you could start to extract those sort of higher order graphs as well. But anyway, cool. I'll be paying close attention.
Excellent. Well, that takes us right to the hour. I wanted to thank you, Lydia and Bing, for demonstrating this great resource and how it's been used. And as a reminder, Lydia will be presenting in the CCF tutorial at 11 o'clock today Pacific time. So you can learn more about how to use this tool as Bing's done in your own work. Next, we'll take a short break and resume with microns and Forrest Collman at 10 past the hour.
As a reminder, we are going to be resuming the program at 10 after 9 Pacific time. That's 8 minutes from now, wherever you are. We're going to have two tutorials today at 11 o'clock Pacific. That's an hour from now. So the information on how to join those is on the login page that you use to find the YouTube and Zoom links to join us here right now. If you need help with that, please let us know. Those two tutorials are going to cover CCF and the Microns Explorer, the resource that we're about to learn about. So we'll post both of those to YouTube. So if you can only make it to one, well, you can only make it to one. They are going to run concurrently. You can watch the other one afterwards as well. Again, the information on how to log into those tutorials is available on the event login page you use to join us here right now. All right, we are going to go ahead and get started again in just another moment. As a reminder, at 11 o'clock Pacific, we will have tutorials on today's topics, the CCF and Microns Explorer. So please do join us then. It's in just about 2 hours. The links to join us are on the page where you got the login information to join us here, or you can find the video on our YouTube channel afterwards. So, Eric.
Thank you so much. It's my pleasure now to introduce the second part of our day today. We're going to transition to one of the newest resources from the Allen Institute for Brain Science, the Microns Explorer. Micron stands for Machine Intelligence from Cortical Networks. And this database includes electron microscopy images, as well as segmentations and 3D reconstructions. We'll be hearing from Forrest Collman, an assistant investigator at the Institute, who'll be presenting an introduction to the database of electron microscopy data. Thank you, Forrest.
Thanks, Eric. Yeah, so it's my pleasure to kind of set up this introduction to this data resource. Today, I'm going to give you a kind of overview about how this data is collected and kind of what this data contains. And then you're going to hear from a number of scientists at the Institute and elsewhere who collaborate with us about different kinds of scientific insights that you can gather from this data. And then in the afternoon in the workshop, you'll hear from Sharmi, Layla, and Casey about how you can really access the data and the sort of nuts and bolts of looking at it and using it in your research. So let's start with kind of reflecting on what is EM connectomics data. And I've got this movie here, which illustrates kind of the classic use case of EM data. You can see there's a rendering of lots of different cells, and there's a couple of neurons in purple and in green. And if we zoom in here on one portion, we can actually see here that this purple neuron is synapsing onto a spine of the green neuron right there. And it's actually also synapsing in this other location, and we can zoom in and find these connections between individual neurons. You can see in gray, there's all these other cells which have also been reconstructed. That's really the joy of electron microscopy, is that when you image something, you really can see absolutely everything that's structurally there within the tissue. Usually the people, when you think of this technique, you think about these large-scale graphs of the brain. It was great to hear Bing starting to think about how she might adapt her techniques to working on these kind of complex graphs. We spent a lot of time thinking about analyzing those graphs. But there's also a lot more that you can learn from data like this. And we picked talks today that in fact highlight that. JoAnn is going to talk to you about glial biology. Carolyn from Genelia will talk about what she's learned about cilia. Nick is going to talk about what you can learn about mitochondrial organization within cells, as well as what you can learn from analyzing the functional connectivity of these data. And Agnes is going to give you a great overview to kind of get you more familiar with looking at anatomical data like this. So I'm going to go on to the next slide. As mentioned, this whole project is funded from the Microns project, which is actually a multiple institution collaboration that the Allen Institute is a part of. And so as an overview, I want to make sure that we kind of highlight all the different teams that have been involved in creating this resource. And it starts in Andreas Tilias's lab at the Baylor College of Medicine, where they have taken a number of all of the samples that were processed through this project were first imaged there in a functional imaging experiment in which 2-photon imaging was used. 2-photon or 3-photon imaging was used to record the activity of neurons. And then that mouse was shipped to the Allen Institute, where it was prepared and imaged for electron microscopy using techniques I'll get into in more detail later. And then finally, that data was shipped to Sebastian Sung's lab at Princeton University, where they applied modern machine learning techniques to be able to automate and speed up the reconstruction of these neural circuits. And then everyone has been collaborating on analyzing the data. And you'll hear from Nick, who is a graduate student in Sebastian's lab at the end in one of these talks this morning. Okay, so this pipeline is extremely complex. There are many steps. This kind of iconograph reflects all these different steps. And I'm going to walk through them in detail, but in kind of classic Allen Institute style, this is a pipeline that is where each step is kind of owned by a particular team or set of individuals. And as I mentioned, it starts with functional imaging and Andreas's lab. Mark is our expert at perfusion and blocking. JoAnn with the histology. We also do micro CT. We have a whole sectioning team to work on taking this block of tissue and cutting it up into thousands of individual nanometer scale sections. We have a large scale microscope that needs to be highly automated that has a team associated with that. Then there's processing all the images, taking all the individual images, putting them back together again, obviously segmenting them, registering them with, say, the CCF, and then, of course, analysis. And you're going to hear a lot about analysis here today, but that's really at the end of the pipeline. And so my job is in part to kind of give you an appreciation about what it takes to get to that analysis phase. And as I said, you'll hear from Casey, Sharmi, and Layla in the afternoon about how you can do your own analysis on this data. All right, so where does it start? It starts with the histology. And today we're going to focus on the data set that's already been made publicly available. That's a data set from layer 2-3 of a P36 mouse. And what you take the brain out of the mouse and you embed it in a hard plastic resin after you fix it. And this kind of iconograph shows you approximately the size of the volume that we're talking about here. It's about 250 microns by 150 microns by 100 microns and located right at the the border between layer 1 and layer 2-3 and going down about 150 microns in the brain. So once you get this chunk of tissue embedded in a hard plastic resin, in order to image it in an electron micrograph, you need to section it up into many ultra thin, tiny sections and put them on a grid, which is transparent to electron micrographs. And this is a video of Agnes actually picking up the sections from this data set in this incredibly difficult, tedious process, which she did heroically, collecting these small ribbons of serial sections, each of which is about 40 nanometers thick, onto this tiny little apparatus, which is a thin film with a section lying on top of it that can be mounted in a transmission electron microscope. Since Agnes did that, we've been working with our collaborators at Harvard on building a sort of more, on implementing their grid tape mechanism, which allows for a more automated section collection. This kind of illustrates the difference between the two techniques. Rather than holding a single grid by hand, there is this film is placed on top of a hole on an automated tape, and the tape rotates through this boat. And here in the movie on the right, you can see the kind of sections happening. Here's the block, and it comes down over a diamond knife, and then a section starts to come off that diamond knife. And this, at the bottom half of the frame, is sort of the rotating tape. And the tape is precisely synchronized with the knife so that as the sections come off, the sections land right on top of the hole with the film. And this is the mechanism now that we've been using to cut tens of thousands of sections from an individual sample. The data I'm going to show you today, and most of what we're going to talk about today, was collected by hand in that video. So once you've got your sections on your ultra thin film, you can transfer them over to an automated TEM pipeline. So we have a TEM pipeline at the Institute that now has 6 microscopes. Each of these microscopes has been fitted with a customized stage and camera system that allows us to take many pictures extremely quickly of this tissue. This movie here illustrates what happens when we're running multiple microscopes in a row. It's about real time. Each microscope runs at about 4 hertz, and there are many running at the same time, each imaging a field of view, and that is pretty large. And so we're able to get up to extremely high data rates with this system. So this is this is the mechanism that we use to build very large data sets. I want to go back, actually. If you want to read more about this high throughput automated system for imaging, we recently published this paper that's in Nature Communications that has a lot of the methodological details in it. So once you've got all of these images of EM, each at high resolution, they have to be kind of put back together again into a coherent volume through a series of image processing steps, which are outlined in this figure that Guthrie made, who is one of the scientists who works on our computational reconstruction pipeline. And so we have 2D images of tiles of EM. The electron microscope produces images which are extremely high resolution, but they have subtle warps due to the electric fields in the microscope that we call lens distortion. So there has to be compensation for that lens distortion, and then montaging the images in 2D to stitch them into a coherent 2D picture. And then each of the 2D pictures have to be lined up in 3D to form this 3D aligned volume. And we collaborate both with Janelia Farm, who helped us set up the software infrastructure that backs this image processing pipeline, as well as Sebastian's lab at Princeton, who really put the final touches on the fine alignment step of the algorithm to make the images really coherent in 3 dimensions. So what are you able to see in one of these images? So here's a kind of medium magnification 2D image of an electron micrograph, and I've highlighted some of the basic components that you can see in this image. So you really see pretty much everything that's there. At a low, sort of low spatial resolution, you can see individual cells. So this red outline highlights the outline of the cytoplasm of a neuron, and this yellow one, its nucleus within it. You can see larger structures like dendrites, which I've highlighted one here in blue. And you can see neurons and non-neuronal cells. You can see this kind of darker nucleus is characteristic of a glial cell, highlighted here in green. Of course, the reason we're doing this is not this kind of medium magnification, but really high resolution. As you zoom way in on an image, you can start to make out the individual wires in the brain. So here on this side, highlighted in yellow, is the bouton of an axon. And then here in purple is a spine on an excitatory dendrite. And this location right here where they meet is a synapse. Pardon me. These little round vesicles here are synaptic vesicles. This is where the neurotransmitter is that will, when this cell fires an action potential, will release their contents into this cleft and signal to receptors on the other side, the very basic mechanism of classical chemical neurotransmission in the brain. So this is a 2D view of an electron micrograph, and you can see there are all millions of lots of wires in this image, and of course, millions of them in the brain. We're interested in reconstructing these in 3D, so we need to take this conceptual step where we've highlighted the individual things in 2D and reconstruct them in 3D. In this video that was produced by Daniel Berger from Jeff Lichtman's lab, another electron microscopy lab, illustrates this process quite beautifully, and so I've borrowed it here to show you. You can see the individual sections, you trace out the outline, and then conceptually you want to do that in every single section, and then form a kind of 3D reconstruction of the object that you're interested in. And you're interested in not just doing this in one object, like it is illustrated in this video here, but multiple objects, and like you'd like to do every object. And so in order to do, oops, I've got a little bit, got the slides a little out of order. In order to do that, you need to really apply modern machine learning methods to take this volume and reconstruct all of the cells and all of the processes that are within the volume. And that's kind of the technique that Sebastian specializes in. Of course, you also need to do one other thing, which is detect the synapse locations. Nick Turner, who you'll hear from later, is in fact the expert at doing this, training machine learning algorithms to identify that there is a synapse here that connects this object to this object. And I just want to further connect the perspective of this 2D view to this 3D view. Here you can see the individual 2D image of electron micrograph overlaid on a 3D image on the right-hand side, in which you can see the full 3D mesh of the axon and the dendrite, and even the 2D slice kind of highlighted in between here. This is a view that you're going to see a lot of today. This is a screenshot from a visualization tool that we use called NeuroGlancer that was developed at Google. And this is kind of how electron microscopists think. They think both in the sort of 2D perspective and then also in this kind of 3D perspective. As I mentioned at the start, though, it's not just neurons that you can see within this brain. Here's a movie of a microglial reconstruction that's adjacent to one particular pyramidal cell. You can follow these purple processes as this microglia makes many, many different contact points with this pyramidal cell. You can see this kind of line that sticks up right there is actually a cilia that you'll hear about from Carolyn. And now you can see right where this microglia contacts the cell, there are in fact many, many different axon fragments that are running underneath that filopodia of this microglia. So this is, what this data is really good for is really seeing absolutely everything that exists ultrastructurally within this tissue. Move on. So to summarize kind of the data that I've told you about, there are kind of multiple data products that you can think about that comes out of this data. There's the 3D EM images that contain a lot of information if you take the time to learn how to interpret them. The project has created this dense voxel-wise segmentation in which each individual compartment gets a unique ID. And from that, you can derive these three-dimensional reconstructions of each of the objects, which are stored in a kind of mesh format. And then there are also synapses, which tell you the locations of where there are electrochemical synapses between objects within the volume. And you'll hear in the afternoon about how you can access each of these data products and use them to perform analysis. All of this data that I've talked to you about today is freely available on this website, micronsexplorer.org, which is a website that we put together in collaboration with our colleagues at Baylor and Princeton that gives you access, links to the data, and describes what the data is, describes who has helped make the data. It also contains links to publications that we've created out of this data. In particular, there are a couple of publications that we've talked about in other forums that we're not going to have time to talk about today, which you can find linked to on this website. One talks about some findings we found about the distribution of synapse sizes amongst layer 2-3 cells that are surprisingly bimodal, which have maybe some implications for mechanisms of learning and memory within layer 2-3 of cortex. And then we have another paper that describes insights that we've learned about the role of chandelier cells and their connectivity within this layer 2-3 data set. So if you're interested in either of those questions, you can find the links to the paper, those preprints on this website. Okay, finally, as was, I hope, clear in my introduction, this effort is a very large team effort, which involves multiple institutions and a very large team here at the Institute and collaborations at a number of other institutions. And so, I'm really proud to be able to be a member of this team and honored that I got a chance to introduce to you guys this data. And I look forward to hearing from the scientists, and I hope you enjoy the workshop later today. And so, of course, finally, I'd like to thank our founder, Paul Allen, for all of his financial financial support and encouragement. So with that, I think we'll take a couple of questions.
Thanks so much. That's really exciting stuff, Forrest. Wanted to field a couple questions. We had one from Kurt Short. Are blood vessels also visible?
They are visible. In fact, one of the very first publications that we have had making use of this data that we didn't really have much to do with was from a biologist at Seattle Children's here who looks at blood vessels. And they are segmented. The segmentation is never perfect in these data sets. There are errors. And one thing I should have mentioned is we've gone to great trouble to proofread as best we can most of the cells that have cell bodies within the volume, as well as some of the axons and dendrites that were of particular interest to us. But there's more work, more proofreading work that definitely needs to be done. And that's definitely true of blood vessels. Some of the blood vessels, because they have particularly astrocytes, they tend to have very close apposition of astrocytes. And so some of the blood vessel reconstructions are incorrectly attached to astrocytes. But you can see the blood vessels. You can see the difference between veins and arteries. There are interesting differences in the kind of ultrastructure that surround these blood vessels. So it's one of the areas of biology that was not the reason we collected the data set, but it's definitely ripe for exploration.
And in terms of that proofreading, what does that consist of?
So when you have one of these 3D reconstructions, there are basically two types of mistakes that can happen in a segmentation. There can be false splits. Say you have an axon that's running along, and then the reconstruction stops, but the axon itself actually continues. And so what should have been one component is now in two or more components. So that's a false split, and you need to kind of annotate and merge those two components together. The other is a false merge, which is what I was talking about with an astrocyte, where say you had an axon and a dendrite that came in very close composition, the membrane got muddy, and the machine learning algorithm got confused and merged that axon to the dendrite incorrectly, say at a spine head, and there you need to kind of separate the axon from the dendrite. This is, Sebastian's lab is one of the world's experts in creating neural networks that deep neural networks that do this in an automated way, but no algorithm is perfect, and so you need to undergo this sort of proofreading procedure to really correct the data and make it as high quality as possible. And for this data set, we've done that for all 300 and I'm going to get the number, I think it's 350 something pyramidal cells within the volume, and the tens of inhibitory cells, and tried to trace out their axons and fix their dendrites so that they are 100% accurate. But there are still millions of other axon fragments and dendrites and things like that have not undergone that process. But you can look through the data yourself. You can click on the link and you'll see how to do that later and very quickly get a sense about what the data quality is.
That's fantastic. We had one more question from Hugo. Are the components of the meshes labeled, for example, as axon, dendrite, Brunton, soma? Yeah, so in we have some derived kind of data products that we make available for some of the cells, for the cells with somas in the volume, in which we have done that labeling, in which the dendrite, soma, and axon components have been labeled, but it's just for a subset of all of the components that are within the volume. So a partial yes. Gotcha. It's definitely another, a project that people work on to build. There are sort of endless sets of derived analytical things that you'd like to have done on this data, such as automatically identify where there are spines, what kind of shape of spine it is, whether it's a dendrite, whether it's an axon. We work on those questions. We also think it's kind of this data is a playground for people who have kind of computational interest to think about how you might formalize and come up with creative solutions to problems like that. Great, and that's a great transition into hearing about some work from other Allen Institute researchers who are working with this database. Agnes Bodor is a scientist in the Allen Institute for Brain Science, and she'll be presenting next about recognizing neuronal cell types in electron microscopy data sets. Really looking forward to this talk and even more amazing pictures and what to do with them. So it's all yours, Agnes.
Hi, thank you so much for the introduction. As it was mentioned, I'm working with the EM team, and today I will introduce how to recognize excitatory and inhibitory cells and even some cell types in the data set. So basically, you can use three types of clues when you're using the data set. 3D clues are available when you're looking the 3D viewer, EM clues when you're looking the electron microscopy sections, and connectivity clues when you analyze the connectivity of cells in the data set. So it requires some work. And so first I'm talking about the 3D gross morphological clues, and I choose this image from a literature because it's a really good cartoon. It's actually represent almost all the clues you will find in the reality of the data set. So you can see here in the middle, a pyramidal cell. And although pyramidal cells also have multiple cell types, they are much more similar to each other than the inhibitory cells. I forgot to mention that the pyramidal cells are the excitatory cells of the cortex, and they usually have a main dendrite, which is the apical dendrite, and it's form a tuft in usually layer 1, not 100%, but almost all of them. And they have basal dendrites, which are pointing sideways or down. And this is why you get this typical triangle shape, both on large view or on a soma area. So probably this is where the name comes from, pyramidal cell. And the axon is always coming from the bottom and pointing down. And this cell types is spiny on the dendritic region. And these spines are the main receiver of the excitatory input for this cell type. When you looking the inhibitory cells, they can have very variable morphology. They can be bipolar or multipolar. They don't have main dendrite. The axon can come out any side of the cell and can point any direction. And they can be spiny and aspiny. And next in a video, I will show you how this is look like in the data set. You can see here several examples for both excitatory or pyramidal cells in green, and for inhibitory cells, which often called interneurons in the cortex, in warm colors as pink, red, or orange. The first thing I would like to emphasize is that the pyramidal cells are much more similar to each other, even if they are from different cortical layers, although here we only have layer 2, 3, than the inhibitory cells, which are very different even within layers. You can see the pyramidal cells almost always have an apical dendrite and many basal, which are going sideways or down. The apical always point up to the pia and usually end in a tuft in layer 1. If you look the inhibitory cells, they can have many or very few dendrites. They can be bipolar or multipolar, and they can be smooth surface or spiny, opposite to the pyramidal cells, which are always spiny. Another structure I would like to mention is the axon, or the axon initial segment, which is the beginning of the axon, which in case of pyramidal cells always coming out from the bottom of the cell, or sometimes as a modification of it, it's coming from one of the basal dendrite, but no matter where it's coming from exactly, it always points down to the white matter of the cortex. Opposite to this, in case of inhibitory cells, axon can start in any area of the soma and can point any direction in the cortex. Here you see an example for an axon coming out from the bottom and pointing down. And here you can see with orange another one coming very, very far from the dendrite and also pointing kind of down a little bit sideways. In the case of these two cells here, the axon coming from the top and then turning backwards. And for this cell, the axon coming from the top also, but it's pointing up. So that's another feature you can use to distinguish inhibitory cells from excitatory. Another structure I would like to show you, which can be very helpful to differentiate pyramidal cells from inhibitory cells, are the spine coverage. You can see every dendrite of the pyramidal cells are covered by spine, although the level of spininess can be different within pyramidal cell types, but all of them have that structure, especially starting on a secondary dendrites. So if you look close, you can see that the morphology is very variable. The most well-known are the mushroom spine with a big head and a neck. You can see here, and another one here. But there are many spines which look like a finger or a philopodia with a much smaller head. But no matter what, they still have a head and a neck. When you look the inhibitory cells, which often called in the literature a spiny or smooth surface cell, actually you will see it's not true. Inhibitory cells can be both spiny and aspiny. If you go close to this cell here, this is a pretty good example for a spiny cell. You can barely see a spine, but if you look carefully, you will still find few, especially on the distal dendritic arbor. But there are many cells which are very spiny in case of inhibitory cell. You can see here a cell with a very spiny dendrite, but if you look close, these spines can have a very variable morphology, and many times it has tiny thorns, and the shape is absolutely not uniform and not resemble the head and a neck structure, as we saw in the case of the excitatory cell. You can see here. So next, I will talk about the electron microscopy clues visible in the EM sections. One will be about the nuclear membrane and another about the synapse structure. So here you can see a cell with a nucleus in the middle, and you can see the nucleus are labeled, the nuclear membrane is labeled with dark gray and the folds with yellow. And here you can see many nuclei 3D reconstructed, and the last column are the inhibitory cells and the others are the excitatory. And you can see that the inhibitory membrane, inhibitory nuclear membrane is much more folded than the excitatory. So this can be again a clue that you can use to differentiate inhibitory cells from excitatory. Next, I would like to talk about the synapse structure, how you can differentiate inhibitory synapses from excitatory. So first, I'm showing this cartoon. You can see the presynaptic terminal. In case of excitatory synapses, the vesicles are much more round. In inhibitory, they can be very variably shaped. And for excitatory, the most common target, not exclusively, but most commonly a spine head. And for inhibitory, the targets are usually dendritic shaft and soma. And you can see that the presynaptic density is thin, while the postsynaptic is thick in case of excitatory synapse, while for the inhibitory synapses, both density are thin. And because for excitatory one is thin and another thick, it's called asymmetric synapse. And for the inhibitory synapse, because both are thin, it's called symmetric synapse. And now again, I'm showing these structures in the 3D, how it's look like in a tissue and in 3D. Electron microscopy datasets contains millions of synapses, as you can see several on this single cutting plane. If you can imagine synapses as little discs, they are placed randomly into the cortex. And when we cut the tissue, we either cut these little discs or synapses perpendicular or parallel or something between. Using this morphological clue to differentiate inhibitory or excitatory synapses is only possible if your cutting angle is closer to perpendicular. First, I would like to show you this example in the middle. Green is the presynaptic terminal with the synaptic vesicles, which are quite round, and it contains a mitochondria. And the postsynaptic structure is a spine head, and all these together already suggests that this synapse is excitatory, since excitatory synapses most commonly found on spine heads. But if you look close, you can see that the presynaptic density is quite thin, while the postsynaptic are deep into the postsynaptic structure. This asymmetry, called asymmetric synapse, suggests an excitatory synapse. There is another clue here. You can see the synapse in two parts, and this called perforated synapse. If you look several sections, or you can look this in 3D somehow, you will see this is one structure, one synapse. It just has a shape that it look like two in given cutting angle. Inhibitory synapses often found on soma or dendritic shaft, and the vesicles are not as round usually as we saw previously. But here also you can see a beautiful perpendicular cut compared to the synapse membrane, but here you can see very thin dark density on both sides. And this is why it's called symmetric synapse. If I show this synapse on few plane, you can see the same. And also compared to my cursor, you can see the synapse don't move much and in XY direction, which suggests a good perpendicular cut, which make this synapse more judgeable regarding inhibitory or excitatory, compared to tangential cuts, where the cutting happened way more parallel, close to parallel to the synapse membrane angle. This kind of cutting plane, which is close to parallel, called tangential, generate an artifact which can look like a synaptic density. Electron microscopy. So next, I will show the connectivity clues using two inhibitory cell types with a very unique connectivity. But first, I would like to mention that we already analyzed several cell types, both excitatory and inhibitory, in this data set or our data sets. And every single one we examined had a unique connectivity pattern. So this is one of the most promising clue for recognizing cell types in data sets. And here is our first unique cell. So in this video, I will show you the chandelier cell or axo-axonic cell, called axo-axonic because the axons of axo-axonic cells target only axon initial segments of pyramidal cell, which are gray here. So you can see these are the axon initial segments, and the colored ones are the axons of the axo-axonic cells, and you can see how they team up and innervate exclusively the axon initial segments. The dendrites are cut off for the pyramidal cell just for visibility. In next, you will see all the pyramidal cells with gray in this data set, and with their axon initial segments pointing down to the white matter, and with red you can see all the axo-axonic cell axon and one of the chandelier or axo-axonic cell also. So if you see axon behaving like this, you know that an axo-axonic belong to an axo-axonic or chandelier cell. Next, I would like to talk about the basket cells, which is it's not one cell type. Multiple cell types belong to this category. And they're unique because they target in a much higher ratio a pyramidal cell soma region, which means soma and proximal dendritic region. From our data set, we made a statistic for one cell, and 26% of the synaptic terminal ended up on soma of pyramidal cells. Here you can see two images from a literature, and the staining are two different basket cell type axon. The green is the parvalbumin positive basket cell axon, and the red is the CB1 cannabinoid 1 receptor positive axon, and you can see they surrounding the putative soma of pyramidal cells, which I labeled with asterisks. And on the last image in my talk, you can see 3 pyramidal cells from the data set, and labeled with blue and green. And you can see with warm color, red, pink, orange, you can see the axons, or actually mostly the synaptic terminals of basket cell axons. And you can see how they surround the soma of pyramidal cell. Probably this is where the name come from, because it's probably look like a basket formed around the soma of pyramidal cells. And this is a very, very strong inhibitory input on pyramidal cell soma. And next, I just would like to say thank you for everybody who contributed in the work and in the Institute, and for you for your attention. And I'm ready for questions.
Great, thanks for the excellent talk, Agnes. I noticed there was a diversity of spines throughout the population. You started off showing us both within a neuron and across neurons. And you sort of touched on this, but did you notice any correlation with presynaptic processes of that particular type of diversity?
Oh, absolutely. Synaptic terminals are very unique, and it also will be a very important clue to recognizing cell types, both shapes, size, synapse number, synapse size, vesicle amount, volume. Every measurement will be important to recognizing cell types, because they are all different for given cell types. Yeah.
And I don't want to get ahead of ourselves, and we'll hear about this, I'm sure, more in the next couple of talks, and especially in the workshop. But can you give us a sense of how easy it is if I see a couple of different types of spines or a neuron or two, how I can go from the just really titillating, incredible qualitative images to quantitative values? Can you give us a sense of that?
Oh, you mean like measure, like
all those values that you just mentioned?
Oh, I think this will be more about the analysis talk or the hands-on, where you can learn about. I personally can only talk about how you can use, like, for example, connectivity clues and stuff like that. I didn't use these detailed measurements, but they are absolutely available, and we're using them as a group.
Gotcha. Excellent. With that, I want to thank Agnes one more time. And our final Allen Institute speaker today, before we go extramural, is JoAnn Buchanan. JoAnn is a senior research associate in the Allen Institute for Brain Science and a PhD candidate at Northeastern University. She will be presenting how a large-scale EM data set led to novel discovery about glia. Thank you, JoAnn.
Thank you for that introduction. I'm really excited to talk to you today and tell you more about how we can use this data to find some very exciting things. So you've heard a lot from Forrest and Agnes about our data set, P36. We also sometimes affectionately call this data set Pinky. However, we'll stick to the scientific term today. And these representations show you've seen before what it looks like, the stack of slices, and also the beautiful 3D renderings. And here you can see some beautiful neurons. But some of you might be thinking, and also Forrest alluded to it, well, what about glia? Well, here in this image, number 3, what I show you is some of the glial cells, known as OPCs, that are rendered. And what you see are that individual cells tile. And what that means is that they maintain their own territories and they don't intermingle with each other. Their branches are actually repulsive. So it's kind of like social distancing, if you will. Now, there is another data set which is not yet public, what will be, and this one is the P49 data set. So it's a little bit older. It's also different because as opposed to the first data set, P36, it's actually long and thin. So what's nice about it is that it includes all 6 layers of the cortex. And what we were able to do looking at that data set was actually to count and quantify and categorize the number of glia. So this chart here will show you the distribution, with astrocytes in blue being the most numerous, microglia in purple second, oligodendrocytes, and finally OPCs. So we're going to take a closer look at the 3D renderings of these type of glia. Here in gold is an oligodendrocyte. Here in pink is the OPC. The blue is the astrocyte, and purple is microglia. I would just like to make a comment now about microglia and point out that they are actually not really glia. Rather, they are blood cells, and they're related to macrophages, a kind of immune cell. The other neat thing about glia, besides the tiling, is that they often can be found in a satellite position. And what that means is that their cell body is closely opposed to that of their host neuron. And this movie here highlights that kind of positioning of the glia. And in this particular cell, this pyramidal neuron in gray, there were actually a microglia and an OPC together in satellite position on this neuron. And you can see how their branches intermingle, and also how one side of the soma is kind of flattened because of that very close relationship with the pyramidal cell neuron. Okay, having a little trouble forwarding here. I'm having some trouble with my slide. Going to the next slide. Thank you. Okay, so now what is an OPC? So OPC stands for oligodendrocyte precursor cell. And these cells are really interesting because they can metamorphose into a myelinating oligodendrocyte, or they can remain in this immature state. They have highly ramified branches that have been shown in live cell imaging to move throughout the parenchyma of the brain and make contact with all kinds of neuronal elements, very similar to microglia. A subset of these cells have specialized endings that encircle synapses and enwrap axons. And as the other glia do, they also tile. But one of the questions has been, since they were discovered, is what else might they do besides a simple replacement of an oligodendrocyte? And because there's many more that are needed, so I just want to tell you a little bit about an oligodendrocyte. So an oligodendrocyte is a myelin-producing cell. And myelin is a fatty protein that surrounds axons and helps to propagate action potentials. Now, there are many diseases like Parkinson's and MS and ALS that you've probably heard of. And demyelination and sort of destruction of myelin by these diseases actually cause the symptoms. So if an oligodendrocyte dies, an OPC can then metamorphose and take its place. And this picture here in green, we can see an oligodendrocyte. In gray is the axon, And a closer look at it shows how this cytoplasmic process enwraps the axon. And here in this beautiful study shows by EM thin section how complicated this wrapping is. And what happens is that the oligodendrocyte sends out these cytoplasmic processes that wrap and form many layers. And the cytoplasm is actually squeezed out and becomes compacted. And then the myelin basic protein just builds up around there, so it ends up looking like this. So I'm going to switch gears a little bit now, and I'm going to tell you about phagocytosis. And phago is a Greek word that means eat. And I bet many of you in Biology 101 have seen phagocytosis either by paramecium or macrophages. And here, this movie shows in red, these are macrophages, blood cells that are phagocytes, and they're being fed thymocytes. And what happens is you can see inside the cytoplasm of the macrophages as the thymocytes get ingested, and then they are eventually broken down and gotten rid of. So phagocytosis is a very important process for maintaining homeostasis. And in the brain, phagocytosis is done by microglia, particularly during development, and they're sort of known as the main phagocytes of the brain. And there are several steps to phagocytosis, and it starts with a signal of find me, because you don't want to eat the wrong thing. So it's really important to know who to eat. And in this case, a dying cell will send out that signal. And what happens then is that the cytoplasm, the branches come out and form pseudopods and actually engulf the material. And this is then brought inside of the cell. And the lysosomes come, which have digestive enzymes, and they join up with the phagosome to eventually become a phagolysosome. And this is important because I'm going to be telling you a lot about phagolysosomes. So in our EM data sets, obviously, EM is a snapshot in time, but we can look at things and in very high resolution begin to understand what we are seeing. And here in this is a branch of an OPC in pink. And the first thing you see is this phagosome. And the phagosome is always membrane bound. And next to that are two lysosomes. And then what will happen is that the phagosome and the lysosome will join together. And then eventually it becomes this amazing looking organelle called the phagolysosome. And this is where everything happens, where all these different things are broken down. Some of them are recycled and some of them digested. And I just want to point out here where this arrow is that within these chambers of this organelle, we are able to see in our data sets what is inside. And right here are these 40 nanometer vesicles. And that was a pretty exciting finding for us to see that. And so what we were able to do was using Neural Glancer, and there are some annotation tools, we could take these cells, the OPCs and the microglia, and just change the opacity a little bit and be able to see the phagolysosomes within their branches. We were also able to annotate and count every single phagolysosome within the cells. And this was very important because we wanted to compare the two cell types. Because the microglia are considered the main phagocytes in the brain and very important, it was rather surprising to see all of these phagolysosomes within the OPCs. And here I just wanted to show you some of the phagolysosomes were actually segmented, and you can see it in 3D. And once again, we're able to see the contents with these vesicles inside of the phagolysosomes. So then that allowed us to count. We counted a total of 36 cells using both data sets, P36 and P49. And we were able to see that particularly in the younger animal, there are so many more of these phagolysosomes. It's very dramatic. And even in the older animal, they're quite abundant. And once again, I just wanted to say how surprising this was and a very exciting finding. So then our next question was, okay, well, what are the OPCs eating? And once again, because of our fantastic data sets and having these 3D renderings available, as well as the thin sections, we were able to see by reconstruction, you could have an entire axon, not an entire, but a long piece of one, and see it. And you see these little tiny collateral branches coming off. And if we zoom in, we're able to see in both the excitatory and inhibitory axon that it's ingested within the cytoplasm of the OPC in pink here and here. So it appears that the OPCs are phagocytosing, and what they are doing is taking little bites out of these collateral branches. So the next question was, well, can we just get a sense of how many they're eating? And so we were able to take 2 different OPCs and just look at one branch, and in every branch, in every ending, look at the phagosome and see what type of axon that was, because it would continue like I showed you over here. And so by going down the length of the axon, looking at the individual synapses, and as Agnes told you, differentiating them from inhibitory or excitatory, we were able to see that they're primarily eating excitatory axons. We also have some RNA-seq data that I won't show you here, but that does demonstrate that the OPCs are indeed eating neurons, and they eat all kinds of neurons. So I would say they're very, very hungry, and they are really helping prune the circuit. I'd like to share this beautiful movie made by Forrest, and another beauty of our data set is be able to share data like this. And what this is one pyramidal neuron, and it is surrounded by 8 glia, 4 microglia, and 4 OPCs. And you're able to distinguish the microglia from the OPCs because the microglia are smaller and they also have much spikier endings, whereas the OPCs are larger, they have a larger territory. But what's interesting is that even though the OPCs won't touch another OPC, the processes of the microglia and the OPCs intermingle, and suggesting that they may have some kind of communication, and maybe they're working together doing phagocytosis, but we don't really know. So, oops. I would just like to conclude with our findings that OPCs are phagocytic. Now, this is a brand new finding, very exciting. It's never been known. It's known that microglia and astrocytes can both phagocytose, but this is a new role for OPCs. And it looks like they're phagocytosing mainly excitatory axons, at least in the cortex, the area we studied. They also express phagocytic genes. And so this shows that they're active partners in circuit refinement. And here again, another picture. In this case, the branch of the OPC is on a pyramidal cell, and it's just taking a little nibble right out of the branch there. And so with that, I would like to thank our team, our amazing EM team. They worked night and day, literally, to make this kind of data. Also our collaborators at Princeton and Baylor, and of course, IARPA for providing funding for this fabulous project. And finally, Paul Allen for his generosity and his great vision. Thank you very much.
Thank you so much, JoAnn. That was pretty great and really surprising. We had a question from Lucas Cheadle. It says, JoAnn, great work. Given the OPCs seem to be engaging in similar phagocytic mechanism as microglia, predominant engulfment of presynaptic excitatory axons, do you think this is complement mediated in OPCs?
Well, our genes, the genes that we've identified in the OPCs are actually different from the microglia, and there might be some immunological complement related thing, but I don't know. We haven't really dug into that aspect of things, but I think trying to figure out now what kind of mechanisms they do use and how different or similar they are to microglia, and even more so, do the microglia work together with the OPCs in performing phagocytosis? I would really like to know that.
That'd be fascinating. And thanks again for that question, Lucas, who is another member of the Next Generation Leadership Council. So thanks for that question. And if anyone else has questions, please post them into the Q&A. I had a hopefully a little bit easier question, but why would OPCs be eating bits of axon rather than the neuron itself taking care of that axon?
Well, I think it's a refinement of the nervous system, of the branches. It's also a way to get rid of excess synapses, which is known to happen during the critical periods in development. And so just by chopping off those extra bits of axon and getting rid of the synapses, it's also possible that they are working, doing something with the axons in preparation for myelination. Because if you think about it, and you have to myelinate an axon and it's studded with synapses, like you have to get rid of those, you want a clean slate to work on. But it definitely also, with the excitatory axons, I think maybe just helping to get rid of the excess synapses. And also microglia have a different time point when they are active, in these critical periods. So I don't think much is known about that. And certainly, again, how the microglia and the OPCs would work together.
Interesting. And along those lines, is there, this is all very new and very exciting. Has anyone looked at the activity of OPCs or microglia in terms of some developmental disorders, for instance, schizophrenia or anything else? Would you think that might be one of the likely many contributing factors to some of these disorders?
Well, it is known that when you get a glial scar, if you have an injury, that all the glia rush there and form this glial scar, and that includes the OPCs, the astrocytes, and the microglia. And so, that's definitely an immune process. And so there are hints that they do work together, but nothing really known in terms of this development and also, these critical periods of how they all orchestrate this and how much they work together.
Great. Well, thank you again, JoAnn. We'll now hear from some researchers who are using this resource for their own work. As a reminder, at the conclusion of the talks today, we'll have 2 tutorials, including one on the Microns Explorer, another on CCF, so you can learn exactly how to access and use this data yourself. Our next speaker is Carolyn Ott. She is a senior scientist at the HHMI Janelia Research Campus, and she will be presenting unprecedented views of visual cortex primary cilia from large TEM volumes provide cell type specific insights. Thank you for presenting today, Carolyn.
Well, I'm very excited. Thank you for the symposium and the opportunity to share what we've been doing. Moving the slides forward here. There we go. So this work is a collaborative effort at the Allen Institute. I've mainly partnered with Russell Torres, and Wei-Cheng Li's lab at Harvard has also been a part of this collaboration. And we're interested in primary cilia in the brain. And I want to start by looking together at the cilium on a neuron. So here, as we zoom in, you can see that in addition to axons and dendrites, there's another process, the primary cilium. There we go. I'm not clicking anything. I'm really not doing anything.
Let's see if we can.
We're going to pull your slides up one more time.
Thank you.
I'm impressed that this is the first hiccup of the day. Much smoother than some other meetings. And thank you again to the whole team at the Allen. This has been really fantastically done. So, and thanks, Carolyn, for giving me that opportunity to thank everyone who made this happen. So let's try it again.
All right. So I want to start with this video. And as we zoom in, you can see the axons and the dendrites that other people have talked about. And I'm going to be telling you about the primary cilium, which you can see here. It projects off the soma. It's not a dendrite or an axon. And it receives sensory input from outside of the cell. So before we go into talking about all of the things we've been exploring in the Microns data set, I want to take a step back and introduce the primary cilium. There we go. So this is a kind of a simplification. Receptors concentrate in the plasma membrane of the primary cilia. When they encounter ligand, they initiate signaling cascades and second messengers enter the cell body, and these can cause changes in things such as transcription, dendrite, outgrowth, excitability, changing the functionality of the cell. And EM is an amazing way to study primary cilia. So here you can see an axoneme, which is the microtubule core, like the support beams of the primary cilium. The basal body is at the base of the primary cilium. This is the centriole that the microtubules grow out of. The plasma membrane of the cilium is continuous with the plasma membrane of the rest of the cell. Oh, I moved my mouse, but I didn't do anything else. I'm going to try to go back using arrows. Here we go. Let's go here. So I'm going to try not to point. So the plasma membrane is continuous, but there's a region at the base called the transition zone that creates a boundary. And the transition zone is important because that's how you get the concentration of receptors and specification of the signals that the primary cilium receives. And primary cilia are performing essential functions in cells. Embryonic deletion of primary cilia is lethal. adult removal of cilia can cause problems like behavior and learning deficits. And there's classes of diseases where people have mutations in proteins, such as this transition zone or targeting proteins that have significant functional issues in the brain. It includes cognitive impairment. The amazing thing about EM is we also get this context where we can see not only the cilium, but everything that's around there. Here I've highlighted a few of the things you've been hearing about: glial processes, axons, and dendrites. And now the primary cilium is a low frequency feature in this complex neuro brain tissue. There's only a single cilium on most cells. And that has made asking some questions difficult in the past. In early EM studies, serial sections didn't capture the entire cilium. You'd get an image like this, but you couldn't see all of the places that the cilium would go. Immunofluorescence strategies that have been used extensively mask everything outside the cilium, and they're dependent on what receptors or proteins are localized to cilia, which can be different in different cell types. So there's no gold standard. So the opportunity to use the microns data has been amazing to let us start to ask some of these questions. And I'm going to start by talking about neurons. So, I want to mention the strategy that we're using. So in the segmented data, it's easy and straightforward to find primary cilia. I have an example here. Segmentation sometimes separates the cilium from the cell body. Oh. And when that happens, I'm going to play this movie. We use the EM to find the cilium. So the yellow arrow is showing you the centrosome that is the basal body. And as I try to play the movie, there we go. You can see as we move in Z, the cilium coming up and out of the cell. So we can use this and then identify the cilium in that way if it's not in the segmented data. There we go. And so let's first start by talking about cilia on neurons. As Agnes talked about, there's excitatory and inhibitory neurons, and I'm showing examples of primary cilia on each of these. And because of the large number of cells in the data set, we're able to compare both within a cell type and between different cell types. I want to show you what some of the excitatory cilia look like. This gallery gives you a little bit of a sense of some of the differences and commonalities between cilia on different cell types. And I'm going to tell you about some of the ways we're measuring and analyzing the data so we can also compare between cell types. So if you think of the cilium as a base to tip, we can create a vector. And that vector length gives us a sense of the environment that the cilium is sampling. And we can see that the excitatory neuron cilia cluster around a vector length of about 7 microns. The inhibitory neurons are average longer, but there's also a little bit of a range. And as Agnes talked about, the inhibitory neurons include several different cell types. And because she and others have identified what some of those cell types are, we can break this out a little further. And what we can see is that some of the diversity we see in vector length also spreads out across these kind of different cell types that we're looking at. And we're also, a vector is not just a distance, but it has directional information. So we're looking at this as well. And here, I've kind of drawn on the left, you can see that there's 2 angles that we're considering for the cilia vector. And the angle that is on the left, the angle phi, is as if you're looking down from the top on the brain and you're trying to see is there any bias in the cilia pointing towards the front of the animals, toward the back, from one side to the other? And we don't see very much bias in this direction. The angle theta, which is on the right column, is assessing whether the cilium is pointing down in towards the brain or out towards the surface of the brain. And as you can see, while there's no commonality with the inhibitory neurons, the pyramidal neurons have a bias that they're pointing out towards the surface of the brain. Now, JoAnn introduced you to glia in this data set. I'm going to talk about OPC cells in a minute, but I'm going to start with astrocytes, microglia, and oligodendrocytes. Astrocytes have primary cilia. We're just beginning to understand the contributions of cilia to signaling in astrocytes. And it's very hard to see the cilium because the intense, the dense network of the astrocyte. So here I'm overlaying that projection image over the astrocyte so you can see. And as I indicated, there are no cilia on the microglia or the oligodendrocytes. And we can look at the glia and make some comparisons to what we've seen in the neurons. Here you can see that the astrocyte cilia vector tends to be shorter than the neuronal cilia. And if we look at the panel on the right, where we're looking at that angle theta, you can see that there's no bias in the angle theta for the astrocytes. These are not generally thought of as polarized cells. But this plot, we've added another dimension, which is adding the vector length and the angle theta and the different cell types. And you can see that the length of the cilium doesn't tend to bias which direction any of the cilia are pointing. Now, I've given you an idea of kind of some of the quantitative, the anatomical comparisons we're doing. I want to show you some of the cilia biology we're looking at as well. So primary cilia can be formed in two different ciliogenesis pathways, and they either form directly from the surface or in an internal pathway in which they end up with a pocket. And we looked at this feature of the cilia on the cells across the volume, and you can see it quantified on the right side. Almost all of the excitatory and inhibitory neurons have cilia that emerge directly from the surface of the plasma membrane, whereas majority of the astrocytes are recessed in a pocket. There's a subset of the astrocytes that are not, and these could represent a kind of subclass of astrocytes, is one of the things we're hypothesizing. Now, if we bring the OPC cells into the equation, JoAnn mentioned that this is a specialized glial cell type, and they do have primary cilia. They tend to be shorter. They have a shorter length as well as a shorter vector. And there's some really fascinating things about the primary cilia and the oligodendrocyte, or the OPC cells. I want to show you here. Here's an OPC cell on the left, and you can barely see the cilium within the little square. So I've blown it up for you. And the top panel is a surface view. And that is showing you that the primary cilium is just barely sticking up out of the cell. Only a small portion is out of the cell. The rest of it is in a large recessed pocket. This is going to have implications for how this cilium perceives signal if the majority of it is masked. These are some of the questions we didn't know we wanted to ask. Is the cilium on OPC cells usually this shielded from signal? Does it change over time? Are there parts, is the cilium growing and shrinking in order to respond? Because there's a little bit of evidence in the field, a few papers indicating that signaling through cilia may be important for differentiation of OPC cells. Now, there's more to the OPC cilia than just this that's interesting. Inside the cilium, because we have the amazing detail of the EM, we can see many vesicles or membrane structures present in these cilia. Now, occasionally, see these in neuronal or astrocyte cilia, and it's only one structure, and it's very rare, but it's much, much more common in the OPC cilia. And so this opens a lot of questions about how that's happening, why that's happening, and what's going on. So hopefully I've given you an idea so far in the first part of how we're looking at the cilium itself and how we're quantifying the cilium. And what I want to do next is tell you how we're thinking about the environment outside the cilium. I've told you that this is a signal receiving organelle, and that environment outside of the cilium has to be having impact on how the signals are, or what signals it's getting. So the first question we're asking is, what kind of processes are even outside near the cilia? And here, as an example, and I won't point to it, but the cilium is blue and it's coming out of the pyramidal neuron as we go through the Z stack. It's a narrow blue marked process there. And I've colored the axons all yellow, and there are 35 different axons that pass adjacent to the cilium. There's 6 dendrites, 2 astrocytes, and a single microglia that come directly adjacent to the cilium. Now, we in the field in general thinks of cilia as receiving bulk transmitted flow, and there are some steps in the sample preparation pathway that end up swelling the processes, so there is more extracellular space than what is shown in the TEM. So we know there is some space for fluid flow, but we're wondering if there's also any evidence for a more directed release of signal onto primary cilia. Now, early on, I mentioned that there's receptors and ligands on neurons. Cilia receptors concentrate receptors for things like neuropeptides, including neuropeptide Y and somatostatin. And so we wanted to look and see, are there dense core vesicles near primary cilium? And here are a couple examples where we can see that there are dense core vesicles proximal to primary cilia. We're in the process of trying to quantitate the occurrence of this versus and interpret that in terms of how likely that is by chance versus something that is directed. But it's at least suggestive that some of the signals cilia perceive could be released in their direct proximity. And there's another source of primary signals that primary cilia have receptors for, and that could be clear synaptic vesicles, which release dopamine and serotonin. And there's specific receptors for each of these that localize to primary cilia. And so we've looked to try to see where are cilia relative to synapses. And as Agnes and Forrest talked about, the synapses have been segmented in the data. Here I'm showing the synapse in a pale orange, and you can see that synapses can pass directly adjacent to primary cilia. I show this for an inhibitory synapse as well as an excitatory synapse on the left side. And we know from other studies that glia that are directly proximal to synapses can detect spillover. And so this anatomy that we're discovering by looking in the microns data set is suggestive that some of the signals cilia receive could be released in this kind of direct adjacency situation. We also see some synapses that are proximal, but not directly adjacent. And in the final panel, I included a synapse that's adjacent to an astrocyte cilium. And again, we're less sure what kind of signals might be released that astrocyte cilia have the receptors to receive. The most amazing thing about this project is the beautiful data and the really terrific people that I've been able to work with. And it's been exciting to learn about primary cilia through the anatomy. And I'm excited to connect with you and answer any questions that you have.
Thanks so much, Carolyn. That was really something else. A whole different side that I really haven't appreciated too much. I was wondering, and you kind of got to this in the talk, for a specialized process like that, which we may not exactly know what the specialization is for. It seemed like most neurons only had one, maybe there was two. There was a lot going on, but I would have sort of expected that there might be more in a cell, or sort of grouped together so that you can get more efficacy out of the processes. Is there any evidence of different sort of expression of these?
So primary and cilia in general are a single cilium. Your rod outer segment is a modified cilium, but there are cells that have multicilia. So olfaction also occurs on cilia, and those cells tend to have a small cluster of cilia, but not as many as something like an ependymal cell, which has many, many cilia. These cilia, primary cilia, don't beat the way a motile cilium does. In the hypothalamus, there's a subset of neurons during puberty that can have 2 to 5 cilia. In this data set, I've seen a couple astrocytes with 2 cilia, but only a single cilium on neurons. And the idea, just like in vision, it's almost like there's a region for capturing information and then sending that information into the brain. So having a single cilium gives you a single decision point, to a single space that you're integrating information across.
That makes a lot of sense. Thanks for that. We have another question from Eliza Eric. Most neuronal cilia were proximal to axons. Do you think that cilia may be involved in regulating axonal signaling? That's a great question.
So there is some evidence that cilia can influence electrical signaling in neurons. In the pyramidal neurons, the cilia are actually proximal to the apical dendrite and at a distance from the axon, which I wonder at some point if that insulates the signaling coming from the cilium versus the axon initial segment. But the volume of the cilium is so small that although there's amplification steps for those signaling, the messages that come in are fairly directed, but they can influence dendrite outgrowth as well as the activity of a neuron. And there's experiments, not by me, but by other people in the field who've explored those things.
Great to hear. Well, thank you again. Gonna move on to almost the conclusion of our program for today. Please remember to join us for the tutorials. I'm really excited about these, which will occur at the top of the hour, which is 11 o'clock on the Pacific Coast. Our final speaker today is Nick Turner. Nick is a PhD candidate advised by Sebastian Sung in the Computer Science Department at Princeton University, and he will be presenting EM circuit reconstruction with functional data and segmented organelles. Thank you, Nick, for this last talk of the day.
Great, yeah, and thank you for that introduction. So hi, I'm Nick Turner from the Sung Lab at Princeton. So earlier, Agnes showed you how to distinguish inhibitory and excitatory cell types in the microns phase 1 data, the public data set. JoAnn showed you the level of detail for studying glia. And Carolyn described how cilia reconstructions give us both interesting insights into how cilia work and some insights into how cell types differ. Clearly, there's impressive cell type diversity and morphological detail within electron microscopy reconstructions. And today, I'll show you other dimensions in the available data for potential analyses and show you a few of the analyses that we've done in the excitatory pyramidal cells so far. Specifically, I'll describe how we've augmented the segmentation and automated synapse detection with an automated mitochondria segmentation as well, and walk through some analyses we've performed correlating these organelles, neuronal morphology, and connectivity. And then I'll describe the functional data available for pyramidal cells in the same tissue and describe the analyses we performed using that as well. People have hit on this a couple times now, but oh, let me go back a bit. See if I can, there we go. So as you can see, in the example section here, there's a lot of information in the EM image, aside from just synapses and neuronal morphology. Mitochondria are actually generally clear to resolve as they're darkly stained globular objects. If you can't see this immediately, that's okay. We designed a kind of convolutional network approach to label the mitochondria throughout each part of the image volume for analysis. And so we can highlight them pretty straightforwardly at this point. What we can then do is associate each organelle with its host cell so that we can capture extremely detailed samples of mitochondrial morphology and distribution throughout each cell within the volume. Here I'm showing one pyramidal cell rendered with each of its mitochondria. Throughout the volume, we have over 2.4 million mitochondria predictions. If you judge the segmentation by overlap in some smaller ground truth volumes, you see fairly good precision and recall metrics, where errors in these scores often mean that a segment is split or merged respectively, not that it's entirely missing or falsely present. Some dendritic mitochondria split and some output doesn't fully cover somatic mitochondria, but overall we feel that this is a fairly good measurement of morphology throughout the volume. There's a lot to see here, and I'll take you through some of the details, but it's worth taking a moment to motivate. Why would we want to study mitochondria within neurons? One reason is that the shapes of mitochondria within neurons seem to relate to mental function. Normal mitochondria taken from the frontal cortex of rats looks like the image above, kind of long, roughly cylindrical shapes. Yet in brain slices of Alzheimer's patients and rat models of Alzheimer's, you often see a different shape called mitochondria on a string morphology. As you can see on the right, the organelle is highly constricted between large bulbous parts. This shape can also occur naturally, yet occurs in much higher numbers in these afflicted cases. Studying how these shapes arise might tell us something about mental function broadly. We actually haven't found examples of this shape within this currently available data set, yet this phenomenon draws a connection between mitochondria and matters that we all kind of care about. In addition, the distribution of mitochondria seems closely connected to the structure of neural circuits. So previous work has used genetic tools to manipulate the diversity of mitochondria, and it turns out doing that also manipulates the spine density within CA1 pyramidal neurons. So both plots here show the results of one such manipulation of a protein called DRP, which is important for mitochondrial fission. The height axis on the left is a measurement of mitochondrial density within the dendrites of CA1 pyramidal neurons, and the axis on the right is a measurement of spine density in those same neurons. There's a clear connection between the two variables within the analogous conditions, as you can kind of tell. Okay, so what is available in the Microns phase 1 data, and how can we use it to learn even more? Well, along with the basic association of each organelle to a host cell, we performed a computational skeletonization of each cell with the soma in the data set. This came up during Forrest's presentation earlier. Here we see the vertices of a skeleton in the dendrite of one cell. A skeleton represents each neurite with a line of vertices like this one. This representation allows us to label each mitochondria with a precise location and spatial extent within each neuron if we associate the organelle to the vertices which are closest to some part of it. Another benefit of using representations like skeletons is that we can share information across analyses. For example, if we label a skeleton to study a cell's axon, dendrite, or soma compartments, we can then propagate those labels to the mitochondria as well. Here we've done such a labeling using a semi-automated approach. The green points represent the cell's axon, the orange are the basal dendrites, the blue represents the soma, and the red represents the apical dendrite and its branches. Combining these two strengths, we get a large and detailed sample of mitochondria for each compartment and can analyze their differences. It's well known that within neurons, dendritic mitochondria are long and tubular, while axonal mitochondria are generally short and punctate. You can see that within this inset here. The dendritic mitochondria are teal and the axonal are red. However, it can be difficult using other methods to analyze the mitochondria in the soma as they're densely packed. Here we can form a basic comparison in 3D using the different compartments of a cell. And we see that some mitochondria are statistically intermediate in size between those of axons and dendrites. I'll note that this is a log scale, so these differences are pretty significant. Birth or neogenesis of mitochondria occurs in the soma. So this suggests that mitochondria undergo a transformation or filtering process to enter the different neuron types, or neurite types, excuse me. We can also draw more direct connections between a cell's mitochondrial density and its connectivity properties than were previously possible. Here we compute a mitochondrial density metric across the cell's basal dendrites using the computational skeleton and correlate it with the density of synapses on the same basal arbor. The left figure describes the density metric, which quantifies the amount of path length in the skeleton that is covered by mitochondria. The right shows a scatter plot relating mitochondrial density and synapse density across basal dendrites, where each point is a separate neuron. This is a more direct comparison than the spine density measurements that I mentioned before. There are several other types of analyses that haven't been performed yet using these data, particularly expanding this analysis to other cell types. But this gives a sense of the kind of depth within the data set, as well as the general approach. Next, I'll describe the functional data taken of the same tissue, as well as some analyses performed by Alex Bay and our group. Before imaging the EM tissue, the team at the Baylor College of Medicine performed 2-photon calcium imaging in a region that overlaps the EM reconstruction. A pink noise stimulus was shown to characterize spacious temporal receptive fields, and oriented stimuli moving in 16 directions were shown to characterize direction and orientation selectivity. One of 30 trials is shown to the right, where each trial had a pseudo-randomized order presenting the oriented stimuli. 112 pyramidal cells of calcium imaging data, and that's also available in all the stuff that we release. The EM reconstruction gives us concrete data on local connections between pyramidal cells. Here is one example pair of cells that are connected by 2 synapses shown within insets on the left. We've done some analysis of these connectivity structures in a few manuscripts, and Forrest referenced those earlier. But there are also a lot of interesting questions one can ask relating this connectivity to functional responses. We chose to focus on the dynamics of a pyramidal cell's functional response. Previous work using optogenetics to study layer 4 V1 pyramidal cells has suggested that local cortical connections act to amplify thalamic input in producing a cell's output. For example, Leanne Scanziani used optogenetics to silence the cortex of mice while they viewed drifting gratings and measured the input using whole cell voltage clamping. They compared the input onto layer 4 pyramidal cells with and without the cortical signal, so just thalamus alone or thalamus plus cortex effectively at a first pass. And they found that the cortical signal provides the major input to these cells, and that the cortical input amplifies the specific tuning of the thalamic output. You can see this qualitatively represented on the right, where the thalamic input is blue and the total input is colored red. The wave pattern in red matches the structure of the blue quite closely, but it's just bigger. We wondered if the same dynamics might apply to the layer 2-3 cells in our data set. Also, within our data, it seemed that a cell's response to its preferred stimulus was quite variable in strength. So it also seemed likely that cells might differ in how they're connected to their amplifiers. Since we have a very concrete look at connectivity, we decided to investigate whether cells with more local inputs produce greater responses to their preferred stimuli. It can be difficult to know whether or not you're capturing the ideal stimulus for a given neuron using functional data. So to start, we decided to test cells for direction and orientation tuning. We factored the response matrix indexed by stimulus direction and trial into components using an SVD, where the first direction-wise component specifies the cell's tuning curve. We then tested for statistical significance of these curves by shuffling responses by direction within trials. If the OSI or DSI score was greater than 95% of the 10,000 permutations we ran, we deemed it significantly tuned. There's one other complication here. The EM volume truncates a significant portion of the dendritic arbor for many of the cells in this first data set. So directly counting the number of connections a cell receives would also bias our measurement. To be clear, we define the number of connections a cell receives to be equal to the number of presynaptic neurons it has, regardless of the number of synapses that constitutes each connection. That's kind of demoed on the left here. To account for this, we decided to use a fairly simple normalization scheme where we divide the number of connections a cell receives by the length of its dendritic arbor within the volume. And that's represented in these blue squigglies here. We call this a cell's in-connection density. If you compute the in-connection density for cells with a significant orientation tuning and correlate that value with each cell's mean response to its preferred stimulus, we see a significant correlation. This suggests that the pyramidal cells that receive more connections from the cells nearby or its neighbors show stronger responses. We also looked a bit more closely at the trial structure to learn a bit more. So the right side of this slide depicts the deconvolved spike train for the 30 trials of one orientation tune cell response to its preferred stimulus. So they're all, each window is the same stimulus, and they're just 30 different trials of closely the same thing. Although we deem the cell to have significant tuning by the permutation test I mentioned before, you can see that the responses are highly variable. We decided to quantify this variable to see if it also relates to connectivity. Go forward. There we go. Specifically, we thresholded the spike trains by a noise value equal to 3 times the standard deviation from the baseline value and counted the trials where the response did not surpass the value. We then defined a metric that we call intermittency, equal to an average of this count across the number of preferred directions of a cell. This accounts for both direction selective and orientation selective cells, which would have 2 directions. Okay. The intermittency value varied a lot through the tune cells, with many tunes being highly intermittent or having sparse responses. Each box on the right shows calcium traces of a single cell to its preferred stimulus across trial, where the intermittency values of these labels lie in the bins labeled on the left. This shows that despite significant tuning towards specific stimuli, some cells respond quite sparsely. Having computed this value, we find a correlation between our in-connection density value and the intermittency, suggesting that cells that respond more have more local connections with their neighbors. Notably, that was not true for the cells that simply receive more synapses across all of their inputs. So we suspect that this effect relates to local connectivity. Overall, the picture that we get from these analyses is that L2-3 pyramidal cells that receive more connections from their neighbors show stronger and more reliable responses overall. Overall, I hope I've given you a sense of the multimodal nature of the data today, that we can extract additional biological information from the detail in the EM and also correlate the structural detail with functional responses. If you'd like more information on these or other analyses, there's a preprint on bioRxiv where we describe the analyses today. There are also other manuscripts out that you should also check out. In this manuscript, we also analyze multiple types of inhibitory cells in the volume and perform a motif analysis of the connectivity graph between pyramidal cells. Let's see. You can also quickly open up a Cloud Jupyter session using our Microns binder repo. This is a GitHub repo of code, but you can also it also contains all the intermediate data used for the analysis I showed today. If you click that button, it'll start a machine in the cloud that allows you to reproduce all the figures. All the work that I've shown you today, including everything, a lot of the stuff in this binder will also be included in the demo right after this talk. Lastly, I'll take a moment to thank my lab, our collaborators at the Baylor College of Medicine, our sources of funding from IARPA, the NIH, and Google, as well as everyone at the Allen Institute and beyond who contributed to this work. Thanks for your time.
Thanks so much, Nick. Excellent job, and excellent job to all of our speakers. I had one question for you, and one that I rue whenever I get this on any of my computational frameworks. You mentioned that you use a fairly simple approach in order to find the, I believe it was the in-connectivity.
The in-connection density, the normalization.
Yeah, sorry, I need more coffee.
A lot of terminology really quickly.
I was wondering if you had tried any other related approaches or anything to sort of benchmark that algorithmic approach.
Yeah, so this was just kind of a first pass to try to account for the cutoff of the volume. We don't take it to be perfect or necessarily like the best way to do exactly this correction, but this was something we knew we needed to do to fix that problem. So that's kind of how we adjusted for it. We did try several different versions of that metric and found pretty similar results across most of them. So we don't think the choice of that metric is the most crucial, but we did try a couple other things.
Great to hear. And yeah, that sounds about right. I didn't know if there was any other questions from the panelists. I know people are eager to get to the workshops. Want to open up to Keith and Tom to help close out the workshop for the day and for the week before we get to the tutorials. Keith, Tom.
Thank you, Eric. Nick, that was a great talk. Thank you very much. It's cool to kind of see all of the different types of science that are going. I guess that was the point of this whole thing, is to sort of see all the different types of science and the breadth of questions that are being addressed using sort of these Allen Institute tools and resources. And again, we want to plug the fact that we have tutorials on how to use all of those resources, and those are also being recorded and will be available on YouTube after the fact. And if you go to YouTube, you can see that we're already racking up a lot of views of both these talks and the tutorials. So hopefully these become a community resource. So Tom, you and I are supposed to close out here. So first point is, again, thank you to everybody for joining this symposium. We're now at the end of the main program. And we have two workshops following this in about 5 minutes. One on the common coordinate framework system that you heard about this morning, and the second on this Microns Explorer. Those will be running concurrently. If you're really inclined to see both, pick one to watch live, get the second one after the fact as it's recorded. Let me just see, we've got a couple other notes here. We hope you're inspired. That's the essence of one of these notes. And then again, thank you to the huge team of people at the Allen Institute who have put on all of the efforts to arrange this, to coordinate this, to put everybody together, to make sure that everything flowed smoothly, to get this streaming on Zoom and then again on YouTube and recorded. And thank you to all of the speakers who have contributed to this. And we hope to see you at future events. Tom, is there anything else that you'd like to add at this point?
Yeah. I mean, I just really want to say, how grateful I think all of us are to the Allen Institute for Brain Science and the team, and really for embracing the open science mindset, which I think really accelerated and has really transformed neuroscience over the past few years. When I started my postdoctoral training, some of these early resources were already available, and that was mainly developmental transcriptomics data set. And so I very fondly remember the days of browsing the websites to try and come up with hypotheses I could then go on and test in the lab. And so I remember my training starting from that data set as a really stepping stone to something bigger and better. And so I really hope that over the past 3 days of this workshop, you've sort of seen some of the evolution and how these resources are both built and interpreted in such methodical and systematic way, and then how some of the external folks and scientists are starting to incorporate those into their research. And I hope it gives you a really nice spectrum of experiences of interacting with this beautiful neuroscience data sets that are being created. So again, thanks everyone for their attention, and hope you get a chance to revisit that and experience the same way as we did.
And I'd like to make one last point here, which sort of follows up on what you were saying, Tom. I think it's worth sort of saying explicitly that when Tom and I came up with the idea for this symposium, one of the motivating factors was the idea that data is ultimately going to be the limiting factor as we move forward in the future. There are a lot of very, very bright people who have great ideas and can write good code and come up with hypotheses, but it's access to data and access to tools that's ultimately going to sort of be separating very successful from those who are just dreamers. And I think that the Allen Institute can address that. And so I went to a small liberal arts college in Maine. That's where I was initially inspired to pursue neuroscience. And at that point, had I had access to Allen Institute tools, I think I would have been able to sort of see myself as engaging in cutting edge forefront neuroscience, even though I was at a sort of a small 4-year institution. And that's sort of the beauty of these tools and these resources and these open workshops and these recordings, that people anywhere, high school kids or current leaders of the field can dive into all of this. And so I want to encourage that and highlight that. Don't feel left out of this. if you're not at some major research institution. That's the whole point of all of this. So with that, again, that's only achievable by something like the Allen Institute. So thank you all for that, and thank you to the community at large for taking part.
Megan, do you have any closing remarks from your end?
No, thank you very much for being excellent moderators. Thanks to all of our speakers and next generation leaders who participated. And we will wrap up now and open the tutorials. Thank you.
Bye, everyone.
Goodbye, everybody. Thank you, everyone.